Hello, and thank you for joining our seqWell webinar today on the plexWell Rapid Single Cell Sequencing Kit. My name is Joe Miller. I am the co-founder and CEO here at seqWell, and I will be today's moderator. We're joined here today by Dr. Simone Picelli, who I will be introducing in another slide, who is going to be presenting on his work using our new single cell product. First, I'd like to take just a moment to introduce our company and our mission a little further. seqWell is a leader in creating genomic solutions that help harness the power of modern sequencing instruments. We specialize in creating products and workflows that help power the next generation of genomics applications. Our plexWell true multiplexing technology is an important foundation for these products, as you will learn, and has helped create scalable solutions to complex genomic problems. You'll be hearing about one of these problems in our newly launched plexWell Rapid Single Cell Kit from Dr. Picelli today. So to introduce Dr. Picelli further, he obtained his Ph.D. in biotechnology from the University of Padova in Italy in 2006 before moving to Stockholm for a postdoc at the Karolinska Institute. After a short spell at the German Cancer Research Center in Heidelberg, he returned to Stockholm in 2012 for a postdoc at the Ludwig Institute for Cancer Research in the group of Richard Stenberg, where he helped develop the SmartSeq2, as well as an in-house version of the TN5 transposase for single-cell library preparation methods. In 2015, he joined the eukaryotic single-cell genomics facility that Richard Stenberg and Sten Linnerson opened at the Science for Life laboratory, also in Stockholm. So we're really pleased to welcome Dr. Picelli today. So with that, again, my pleasure to introduce Dr. Simone Picelli, and we are really looking forward to hearing about his work here today. Simone?
Yep. Thanks, Joe, for the introduction. Welcome, everybody. I will start my presentation by giving you a quick overview of the single cell field. So you are probably familiar with the 10x Genomics technology. The 10x Genomics technology is a tag-based method that is carried out in emulsion droplets, and it's aimed to characterize only a portion of the RNA transcript, the 3' end. There are several of these methods, both characterizing the 5' end or the 3' end of the mRNAs. But what I'm going to talk about today is are the full-length methods, which are covering the entire length of the transcript. And among them, the most popular, let's say, is the SmartSeq, with the SmartSeq2, which I developed during my time in Stockholm. And now there is a new version of SmartSeq method, which is the SmartSeq3, which was published by the group of Richard Stenberg last year. So a question that many people have is when to use a full-length method or a tag-based method. And then the question is, the answer is, of course, it depends. In the beginning, when we want to carry out an unbiased analysis, it's probably better to choose a high throughput method like with 10x, because the cost per cell is lower. But once we have a more specific question, we can analyze subpopulations, for example. If we have a marker available, we can isolate these subpopulations with the fax or another single cell dispenser and do a full-length method, which is like all the SmartSeq methods are plate-based. And therefore, they have a lower throughput than emulsion-based methods, but they can capture the entire complexity of the transcript, of the transcriptome. So they are able to characterize SNPs, distinguish between splicing isoforms, transcriptional start sites, and so on. And of course, they have a much higher sensitivity. So than emulsion-based methods. They have actually the highest sensitivity among the single-cell methods, and they are considered therefore the gold standard in the field. SmartSeq2, for example, was used in the Human Cell Atlas exactly for this reason. And as I said before, they are useful, or they are ideal when we have a smaller population we want to study. Unfortunately, there are limitations. And one of the limitations is that despite the high sensitivity, the number of genes that we can detect in each cell is still relatively low. And this is due to the fact that the enzymes and the chemistry of the reactions is suboptimal. So we have tried here to improve this or to at least address some of these limitations by using new buffers and new additives. Another problem is that plate-based methods are time-consuming because the cells have to be kept in separate wells of a plate until the very end when there is when they are pulled together before the sequencing, because the indexing of the cells is done very late. In the single cell rapid method, we have found some solutions that are making the protocol clearly faster compared to, for example, SMART-seq2 or SMART-seq3. We also looked into the miniaturization to see if it's possible to scale down the reaction volumes in order to scale down the cost and hopefully improve the performance. I will quickly go through the SMART-seq2 workflow. SMART-seq3 is very similar. There are just a couple of modifications that I'm not addressing here for lack of time. But all these SMART-seq methods and also the single cell rapid method start with the position of a single cell in a plate by using a fax or a single cell dispenser. And the plate contains a lysis buffer, so the cells are lysed upon reaching the lysis buffer. And there is also an RNase inhibitor to prevent the RNases and prevent RNase degradation. In this lysis buffer, there is also an oligo-DT, and this oligo-DT carries a known sequence at the 5' end, which is shown in green here. And the oligo-DT is used to reverse transcribe all the polyadenylated RNA species in the cell. So the first reaction of the protocol is a reverse transcription, where reverse transcriptase is generating a cDNA complementary copy of the RNA, and upon reaching the 5' end of the mRNA is adding some untemplated nucleotides between 2 and 5, and generally cytosines. In the reverse transcription mix, we also have a second oligo, which is a template switching oligo, which has the same sequence at the 5' end as the oligo-DT, and ends with the 3 riboguanosine or guanosine analogs. And the purpose here is to allow the annealing of the TSO to this tail of cytosines. So the reverse transcriptase can continue and make a complementary copy of the TSO. So as you have probably understood, in the beginning, the reverse transcriptase is making a cDNA using the RNA as a template, and in the end is making a cDNA using the TSO, which is DNA oligo, as a template. So it's switching template between RNA and DNA. But the end result is that after RT, we have amplified all the RNA molecules in the cell, and now they all carry the same sequence at both ends, which also means we can amplify them in a suppression PCR reaction in order to increase the amount of cDNA that is available. After cleanup, we have very long cDNA molecules that need to be, of course, fragmented because we are using Illumina sequencer for sequencing. And a very effective method to do that is the tagmentation that is using the TM5 transposase. So the tagmentation is fragmenting the DNA in a 5 minutes reaction, and at the same time adding adapters at both ends. Then there is a final amplification step where we also add index adapters to each well of the plate, and this is just for multiplexing purposes and to decrease the sequencing cost. The tagmentation can be done in several ways. One way is to use the Nextera XT kit from Illumina. Another way is to use the plexWell kit from seqWell, or for the labs that have a possibility to just synthesize its own TF5 transposase, as we published a few years ago. So single-cell rapid workflow, as I said, is very similar to SmartSeq2, but why is it faster than all the other methods? And the reason is that there are a couple of key changes that we have introduced. One is that we have combined RT and PCR together in a single step. We have shortened the RT, which is now only 30 minutes. There is no RT inactivation step, so we go straight into PCR. And since the chemistry has been improved, we also need fewer PCR cycles to get enough cDNA for the tagmentation. If we couple this first part with the plexWell library preparation kit, which also requires less hands-on time compared to other tagmentation protocols, then you can appreciate how faster the protocol becomes. We compared several methods in this study, and we used also several sources of material. We started with the picogram levels of total RNA, and then we switched to HEC293 cells. But in the end, to benchmark a protocol, we needed some challenging cells. And for challenging cells, I mean cells that contain very little RNA. So a perfect example are human peripheral blood mononuclear cells, or PBMCs, which contain 0.1 picogram or sometimes less RNA per cell. So we isolated all the PBMCs from blood by using CD45 antibodies against CD45 surface marker, and CD45 is a pan-leukocyte marker that is present on the surface of all the leukocytes, and it was chosen in order to introduce the least possible bias in this experiment. We did single-cell sorting by using a FACS area fusion in both in 96-well plates containing 4 to 5 microliters of lysis buffer depending on the protocol, and later in 384-well plates containing 1 microliter of lysis buffer. And I will refer to this as a miniaturization experiments. We dispensed all the reagents with IDOT, which is our nano dispenser, and steps that could not be carried out with the IDOT were done with the Fluent liquid handling robot from Tican. We benchmarked 4 different methods, SmartSeq2 according to an updated version of the protocol, SmartSeq3 by using the protocol published in Nature Biotechnology, as well as the latest changes available on protocols.io. And we tested the latest SmartSeq single-cell kit from Takara Bio. Here, for cost reasons, we reduced the volumes by a factor of 2. I think that Takara doesn't mind. The miniaturized protocol should actually work better than the standard volume. And we tested the single-cell rapid kit from seqWell. First, we did an experiment in 96-well plate, and then we also did a miniaturization experiment in 384 well plates. So as I said, we isolated the PBMCs and sorted them in 96 or 384 well plates containing lysis buffer. Lysis buffer was dispensed with the IDOT, and the plates were stored in the freezer or until we were ready with the sorting. And after sorting, they were put back at -80 or processed immediately. Again, with the IDOT, we dispensed the RT-PCR mix, and then after that, with the Fluent, with the Tecan Fluent, we did a magnetic bead cleanup to check the cDNA quality. So we checked for the size of the cDNA on the bioanalyzer, and then we quantified every cell with the picogreen. The next step was to normalize the cDNA concentration in all the wells, even though with the plexWell kit that was not really necessary, because it's quite tolerant in terms of cDNA input, but since we were preparing the plates also for doing the library preparation with an Xterra XT kit, we normalized everything, and we took 150 picograms for fragmentation, and we processed SMART-Seq2, SMART-Seq3, and Takara, and the cells done with the Takara kit, with an Xterra XT kit, and the single cell rapid cells processed with the single cell rapid kit were also prepared with the plexWell library kit. In the end, after indexing PCR, we pulled everything and we sequenced on the same flow cell of an XSeq 550 with a 75 base pair single end mode. And these are some of the statistics. It's really nice to see that the single cell rapid had a very high percentage of uniquely mapping reads, which was very comparable with the Takara kit and better than SMART-Seq2 and SMART-Seq3. Also, the miniaturized version of the rapid kit was performing exactly the same, even nicer probably, because the distribution was much tighter. We then looked at how many genes we could detect with the different methods, and it was interesting to see how the single cell rapid is just detecting a bit fewer genes than the Takara kit, which is the current market leader in the field, but clearly more than SMART-Seq2 and also more than SMART-Seq3. We then looked at the downsampling effects on gene detection, because all these cells from different methods were sequenced at different depths. So to correct for these inevitable differences in the number of genes we can detect based on the sequencing depth, we performed downsampling at 50,000, 100,000, 250,000, and 500,000 reads. And as you can see, SMART-Seq2 is low and stays low. SMART-Seq3 improves with increasing number of reads, and single cell rapid and Takara are performing similarly. Takara is always a bit better, of course, but this, on the other hand, this was the first version of the single cell rapid kit, so there is a room for improvement. But the only, let's say, improvement that we could introduce with this first version was, again, trying to miniaturize the reaction. And once we miniaturize the reaction, we can see there is a clear improvement in the number of genes we can detect, up to 25% more. which is important because in the end we would like to maximize the number of genes we can detect and try to sequence, of course, as shallow as possible, since the sequencing costs are taking up most of the budget when you do single cell sequencing experiment. We also looked at reads that are let's say unwanted, or that they are not relevant for most of the studies, which are we look at how many reads are mapping to the ribosome or to the mitochondria, and these are an indication also of cells not being healthy if we have a lot of mitochondrial reads, and the ribosomes, of course, ribosomal reads are a problem because the ribosomal RNA represents 80 to 90% of the total RNA in one cell. And even if we are using an oligo-DT primer to prime only the polyadenylated RNA, there is always a certain percentage of ribosomal reads in the library. But as you can see here, the single cell rapid has a very low percentage of ribosomal reads, which are less than 2%. And also with the miniaturization, this percentage doesn't increase so much. And the same here for the mitochondrial reads. We have a bit more, but they are around 5%, which is perfectly acceptable. We then look at the gene body coverage. So ideally, we would like to have a method that is covering each gene in a uniform way, independently of the gene length. But that is, of course, a perfect scenario that doesn't happen. Also because we are using an oligo-DT primer, so we start the reverse transcription from the 3' end. But it was nice to see that the gene body coverage is fairly uniform, and then is comparable to the Takara single cell kit. This unusual distribution for SMART-SIG3 is perfectly normal, is just due to the fact how the method is designed. We then look at the reads, where the reads are mapping along the gene. Are they all specific and mapping to coding sequence exons, or do we have a lot of intronic reads, or is there 3 or 5' bias? And as you can see in the graph on the upper left, the reads are mostly exonic in the coding region, both for a single cell rapid and the Takara kit. And while the amount of reads mapping to introns, which is the next graph, is also very low. SMART-SIG3 has overrepresentation of intronic reads for reasons that we don't understand and we couldn't explain. But otherwise, it's the statistics are very comparable between the single cell rapid and the Takara kit. And with this, I want to just finish and summarize our results. We could show that single cell rapid has a very high sensitivity in terms of number of detected genes, higher than SMART-SIG2 and SMART-SIG3, regardless of the sequencing depth. Once we miniaturize the experiment, we could detect even more genes, up to 25% more, which makes a single cell rapid the method with the highest sensitivity among all the commercial and non-commercial methods that we have tested here. And we also shows that single cell rapid has a very high percentage of uniquely mapping reads, which combined with the high sensitivity helps reducing the sequencing cost, because we don't need to we are sequencing less unspecific reads like ribosomal or mitochondrial, or we are wasting fewer reads, and let's say. And we also could show that the kit has gives a very even gene body coverage with, as I said, a lower percentage of genes mapping to undesired portion of the genome, and also very low number of reads mapping into intergenic regions. And the combination of the single cell, let's say, using the full, so using the single cell rapid for generating cDNA, and the plexWell library preparation kit, and miniaturizing with or without miniaturization. In any case, there is a significant cost reduction compared to the current commercial kits. But the major advantage here is that the protocol is faster and is easy to automate and can be completed within 1 day. Actually, 6 hours takes from cell sorting to finally library ready for sequencing. And with this, I want to thank all the people who were involved in this experiment, in this study. In my group, Vincent, who did all the bioinformatic analysis, and Dinko for the great help in the lab. And then from Boton Roska group, Boton Roska is the IOB director, and I got quite some help from Tiago with the facts, and of course our collaborators at seqWell. And thanks for your attention.
Thank you, Simone. That was a great talk. Thank you very much for sharing your results. So I'm going to, we're going to take a few questions here from the audience. So the first question, and I think we'll send this to Simone, is from one of our participants today, which is how well does this work with random primed RT in order to obtain 5' ends and get transcripts lacking significant or any poly A tails? Simone, would you like to weigh in on that?
We haven't done it, but we are of course thinking about it, and yeah, it should work, but we haven't tested yet. I don't have any. Yeah,
I think it's fair to say, yeah, we that is not a supported application of the current kit, and as you probably gathered from Simone's work. I guess, yeah, in terms of the workflow, Simone, maybe you can just again address maybe the question of how one guarantees or knows that it's a single cell when you're analyzing the data, or when you're going through the workflow. Can you talk about that a little bit?
We make sure already in the sorting that we have single cells, and yeah, so we have a way in the sorting to exclude the doublets, but of course it's not 100% sure that we have single cells in the plate, but it's can also be inferred. Well, first of all, we are using a CD45 marker. So if we would have 2 different cell types in the same well, then we would be able to see that. We can also see it by looking simply at the cDNA yield after amplification. If it is much higher, it's most likely coming from 2 cells, and we could exclude or monitor and watch this well specifically, and this is what we can do basically.
Yeah,
but they are very few. If the cell suspension is prepared in the proper way and every step is done properly, the chance is very low.
I mean, maybe related to that, I guess, is there a number of cells per well factor here again for wells that may have more than one cell? Have you looked at the performance when there's more than one cell per well, or is there anything in your mind that on that front that would relate to the anticipated performance here?
Well, if there is more than one cell, generally the cDNA yield is clearly much higher. So can be, of course, like a large cell, but it's a really outlier by a factor of 2 or 3, for example.
Yes, I think that for the next question, again, question from the audience, which I'll answer. The question is, does this single cell rapid kit require FACS sorting into individual wells? So I think as you probably gathered from today's talk, this is definitely a sorted cell workflow, for just work downstream of either FACS or cell dispensing platforms. Maybe again, Simone, a little bit on the performance observations that you had. One of the questions we have here is why the lower volume lysis buffer produces higher sensitivity. In fact, I think there's more than one question that sort of relates to that. Do you have a sense for why that may be the case?
It's an observation that has been done since many years.
Right.
I don't have an explanation, but it's probably the reaction kinetic is better in smaller volume, up to a point, of course. Because if you reduce the volume too much, then you run into another problem, which is evaporation issues. And that when you do an experiment in plates, that becomes a problem. But the miniaturization here, we did a FACS sorting in 1 microliter, but then we added 4 microliters of RT-PCR mix. So in the end, yes, it is miniaturized 5 times, but the volumes are still relatively large. And it's the lower the lower the volume for the RT or PCR mix, the higher the risk that you run into evaporation issues, and that depends on the protocol. So, for example, depends on the ratio of RT mix compared to the lysis buffer. So in SmartSeq3, for example, there would be much higher risk because the amount of RT mix is very low compared to the lysis mix, and with the Takara as well. So it's not easy to generalize, but in the end, smaller reactions always seem to work better, up to a point.
Perhaps a related question to that, again, kind of along that same line of the efficiency. Do you have any sense, again, with your experience here, on what your estimate of the transcript capture rate is? The percentage, let's say, of transcripts that would be typically captured in this type of experiment, and then more specifically, I guess, in this data.
No, we haven't looked into that. We know that SmartSeq2 had a capture rate of estimated between 30 and 35%, and here you can see that is much higher. So with the PBMCs, we generally captured, I don't know, between 1,000 and maximum 2,000 genes per cell, and here we can go up to 3,000-4,000. So it's I'm not saying that it's 80%, but it's significantly higher.
Sure. I guess related to that, again, from a kind of sort of typical use scenario, what in your experience, what would you recommend a user of this workflow, and use in terms of recommended sequencing depth or coverage? The type of read configuration. Again, do you have any thoughts on what the sort of necessary level of sequencing coverage would be for experiment with the workflow that you walked through today?
It's difficult to generalize, depends on the study, but we didn't see any benefits in sequencing. For example, I mean, we are doing a single end sequencing because it's cheaper and is sufficient, and we generally aim for 500,000 reads per cell, but it depends. I mean, when we are now doing this protocol benchmarking, having 1 million reads per cell helps, but it depends. If one wants to process many plates, maybe even 250,000 reads per cell would be just okay to have an idea.
Great.
The budget is the limit.
I think maybe just one or two more questions here. I think one question that was asked a couple of times by our audience is, whether the kit is either compatible with UMIs or includes UMIs. I'll answer that. From the view of what the product contains. It does not come with a UMI sort of barcode built into the RT and amplification reaction. It's certainly something that I think is very interesting, and it would certainly be, I think, an interesting way of enhancing a kit of this type. Another question, again, in terms of again, things that we have not yet validated or looked at is, have we looked at RNA DNA hybrids instead of amplified cDNA? Again, I think another very interesting question. I know there's been a lot of work to look at such methods. This method uses, does use amplified cDNA as its input. I don't think we have too many other questions here. I think we've covered a lot of the major points of these questions. Maybe one last one, Simone, just in terms of again, performance here, again, fresh versus frozen. I realize you probably haven't exhaustively characterized that, but maybe just kind of answering that maybe a little bit more broadly, for plate-based workflows. Again, how do you think about the performance differences kind of anticipated coming from fresh cells versus frozen cells for a workflow of this type?
We haven't compared them side by side, but we have done other experiments that I didn't show here, where we both use frozen PBMCs, and actually here I think that there were also some frozen PBMCs. And we also, with hex cells, fresh and frozen, there is, we use a live dead cell marker, and we sort only the CD45 positive and propidium iodide negative, so we are sure that we are sorting only the healthy cells, and we didn't see any major impact of the freezing.
Thanks. Yeah, I guess one last question, and again, this is a kind of more forward-looking, but maybe your perspective, sort of from a vision, of where the single-cell genomics field is going. Where does your view on what the real kind of critical next advances in single-cell genomics is? And again, maybe how does that, in your mind, connect to routine use in the clinical environment, perhaps. But yeah, just curious for your view on kind of what the the real sort of in your mind the next leaps in single cell genomics is or should be.
For sure, being able to do a multi-omic RNA and surface protein, intracellular proteins, because in the end we can look at the RNA, but the real effectors are the proteins and chromatin. This, of course, with the higher throughput, ideally. So plate-based methods are still an edge because they have a higher sensitivity, but they have a relatively low throughput, but it depends on the question. But I would say I would like to have a method that is sampling RNA, protein, and chromatin and methylation
Sure.
at a high throughput, lower cost, fast.
Yeah. I think that is certainly top of the list for many people. I think yeah. So I think that's I think all the questions that we're probably going to have here today. I know there may be some of you who didn't get your question answered. I would encourage you again, if we did not get to your question today, please feel free to visit our website and reach out to us there. You can email us directly at info@seqWell.com, and we're glad to follow up with you there as well. We really appreciate everybody's feedback and again participation in the questions today. And again, I want to really take a moment here to thank Dr. Picelli for presenting his work using our new plexWell rapid single cell kit. And again, thank all of you for your attendance. As we wrap up, I just want to remind you that this webinar was recorded. We will be emailing all of the participants a link to the recording that you will be able to access in the upcoming days. Please stay tuned. We will have more webinars to come and further product launch details that we're very, very excited to share with you. And so, yeah, I guess with that, we will close. Again, thank all of you. Thank you, Simone, and we appreciate everybody's attendance at today's webinar. Goodbye and take care.